Hey guys, Mr. Dr. Logic here. I know I said I wouldn't be doing more of these homework videos, but I couldn't help resist. I wanted to see how far this would go, and I wanted to break my milestone of 100 subs of this channel before 2022. If we can make that a reality, that would be amazing. Also, please like, share, and subscribe. This really helps this video and this content grow. It'll maybe motivate me to do more of this, but yeah. Also, I'm going to show you this cool other thing. Look at this. You go to view, like all the way up here, view, focus mode. That looks incredible. And then you could play around on this just as well. This is so amazing. And in order to get out, you click this button, not the X. X completely gets rid of it. Or you could do this. Oh, I just found dark version. I just found dark version. We're learning as we go, people. We're learning as we go. But yeah, this is so amazing. Okay, so anyways, I'm going to continue doing it like this so that there are no more distractions and we can continue on our way. So yeah, this is probably going to be the last homework. There is a lab after this that's due next week, and then the special assignments. I'm not going to do the lab or show a video about the lab, but I will do a special assignment. I might finish it soon, but the video will come out a lot later. Also, quick note, I was about to say someone else, but E.coli, like our professor, he did like a walkthrough video of it. And if there's still any more questions, I don't think it warrants a video on its own, but if there are any questions, just let me know about it. Okay, so question 1. Literally draw the efferent. Make sure you draw this. It's up to you if you want to draw the regulatory gene, but do not draw it. I basically copy pasted this image because I know some of you all will try and snip this from the YouTube thing. Nice try, smart asses. I've also gotten copyright stricken because some people were upset from copying from me. So I'm not gonna allow that to happen. So yeah, basically draw the cap site, which is important, the P, this green P, and then the operator. It's got to be like OAUX, and then in between the lacY, another OUX, and then you can just leave it at that. It's not clear if we should do more of this, but basically more or less looks like this, and then after that specifically, because it's meant to close the lacY. That's why those two are important. And as we go down further, that's how it'll be. Also, this question was a bitch. Drawing this was a bitch. I just basically went on paint, drew a few lines. I think you needed how many? 1, 2, 3, 4, 5, 6, 7, 8. 8 boxes. You need 8 boxes. Okay, and then just color code it efficiently, label them, call it the thing, copy paste, dump it here. That's important, okay? I would edit this, but I'm too lazy. This is just more reference. This is just to further strengthen my point that the lacY, I'm calling it the lacY, it's like lactose, whatever. That's the thing that needs to get bound up like a knot or like a, I was about to call it a tennis ball, but that looks nothing like a tennis ball. Also, these purple things look weird. I know what it is, but it looks weird. Couldn't they have drawn like a better picture? It's like a violin grow legs. Nasty. Okay, question 2. Okay, the role of the like blah 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 blah. Okay, so basically everything highlighted is important to address. I wouldn't say address, but basically gets the point across. So again, promoting that cleaves the lactase. It's negative. You got to mention that it's negative. Negative repressor of the thing. Naps on the lactose. This thing binds to it, and then if we go further up, this is what happens, and then it causes that. Well, that's question 3, but this goes up, binds to it, and sort of tries to loop it around. That's question 3 on the specifics of how, but basically negative, bad, no gene expression. If we go to question 3, yeah, like I said, more about the specifics. Basically making a loop. If you don't want your fingers to work, just just cross it. that's basically question 3. Just address how the protein binds to the OAUX and the O main, and basically this is the lacZ. Has to be, because I'm pretty sure it does also for lacY and lacA, but they never make that clear. Based on this image alone, you could claim ignorance, and based on a lot of other images on like Google Images, this is how they portray. They don't even show the O main or OAUX. I don't know how it does it on Smart Bio. I forgot completely, but yeah. Question 4. Uh, I forget. Oh yeah, back to question 3. Negative. Question 4. What role does the thing? It's a promoter. It's a promoter. Well, as you're gonna see why later, but in the presence of. Oh, this feels so nice. This is my first time learning more about this. Okay, so you can see here it converts it into lactose into alactose. I'll get into why this is important later. It binds to a repressor. Basically, the importance of the repressor is to prevent the thing from spending too much energy doing unnecessary work. That's why we have repressors, or at least E. coli in this example, has repressors to prevent itself from developing mRNA and then mRNA to protein, which is meant to specifically break down lactose. But if that lactose isn't there, it's just wasted because mRNA is very unstable and it'll be destroyed almost instantaneously. So it makes alactase. I don't know what difference does it make. I don't care. And then from there, you can see the RNA polymerase. There's more to it than that. Transcription, and then it goes to this mRNA. Then it makes this beta whatever. And then from there, as we mentioned earlier, it converts lactose into alactase. And this specifically binds to the enzyme, changing the violence shape and making it impossible for it to bind on the and the lac main. So this basically says, don't get in the way. We have what we need. Stay out of this. Address it this way and be sure to make sure you say that it promotes. Doesn't support regulation. It promotes. It doesn't. I was about to say supports regulation. It promotes, and it doesn't support regulation. Okay. Next, what is the purpose of lacY gene? There wasn't much on this on the sheet, even in Smart Bios, I believe, but more or less it says it encodes for the permease, and then this brings it in. It's one of the 3 products done by the mRNA. So there it is. And so this basically allows entry for lactose in the body. That's all there is to it. There's not much. Honestly, this is more of an intimidation question. Okay, this is more to say, did you read it? Are you paying attention? I thought it was more or less an easy softball, but if they wanted it to be easy, they would have given us 5 questions. This doesn't need to be thrown in the mix. This is just bullshittery. Question 6. Okay, I basically did like a copy search or copy paste, but in the article they didn't give too good of a definition. It was very confusing, so I googled it. Nothing against that. So basically, it's like the expression level default of mRNA per RNA. This basically summarizes it perfectly. It essentially how much cell or tissue produces. I'm not gonna read. Just you can read this for yourself. It's essentially how much that cell or tissue produces a given mRNA or protein during normal circumstances. I left flora level and ceiling level there because it determines how much or how little you can make. So yeah, that's what basal level means. There were a few references inside the article, but it didn't make much sense. So I just left it at that. This more or less supports lacY more than it does anything else. But this is important. The article doesn't touch upon it as importantly as it should. This is more or less like a baseline saying that this is how we say something can produce something or not. Okay, going into everyone's favorite, question 7. Now this one is pretty ass, okay? Long and short, if you wanted to answer this immediately, what role does it play in? It basically is responsible for the promotion of mRNA. And like I said about basal levels earlier, how much of it? So yeah, it binds to cap, and then the cap helps recruit the RNA polymerase multiple times if need be. And then you can see 3 scenarios here. This is correct. I just ripped this straight off the article. Besides, everything else that you see highlighted and everything, straight out of the article. You can look these specific words up and you can find it immediately. So, more or less, this essentially says if glucose is present, CAMP is low, and there's no lactase. So this repressor will bind. Nothing mentioned about the repressor at all. It just believe in believing. Okay, so this repressor exists, and it'll just bind to the operator if there is no oppressor. And what takes out the repressor is the lactase. And this is what this suggests. Glucose is still there, and lactase is there to prevent the repressor from actually coming down and just saying, hey, no, stop. Basically, you're stopping the stopper. You Uno reverse card the literal moon. But in B, you get very little mRNA because the cAMP cannot, there's very little of it because glucose is present. There's no explanation why this is the case, but just believe it. And then lastly, there's this third scenario where it's like, no glucose is present. I don't know how that's even possible, but cAMP is high. I think glucose reacts with cAMP, but I'm not going to suggest anything. Lactose is present, and there we go, abundant mRNA. And this table highlights it perfectly. And yeah, this is just more for the proof about me just yelling, it's present, thus. That's all you could find about it. That's honestly a load of shit. It might be extra detailed, but I wanted to know more. I looked it up on Google, nothing. Pass. Okay. Lastly, question 8. This is the final question, okay? This one was just straight up a pain. Like, you don't know how to even answer a question like this. I gave it my best shot. It's what you see here. How can something so tiny, such as a purine, basically anything in DNA, affect or regulate the gene? Fuck that, man. Okay, so basically, it is important because we've seen this both in prokaryotes and eukaryotes. I don't know if that's important to mention, but I didn't highlight it. Where is it? Very common in bacteria. It's found in both prokaryotes and eukaryotes. I know I didn't highlight it, but give me a break. Gene expression is modulated by these little fuckers. You can find like them binding to short stretches of mRNA in small molecules. They change the configuration greatly. And so what it does is this riboswitch, I thought it was roboswitch, and I read it roboswitch for a lot, binds to the target molecule and inhibits nearby transcription. Because, I wonder why. So that we don't have two blobs against each other. Whatever. Okay, so to date, yeah, yada yada yada. It's very important because it affects all these three things. And we know it's existed for a very long time because it doesn't require much factorization. How does it regulate it? I guess it inhibits nearby transcription, but like I said, there's not much to go off with what the article gives, and that's why I hated this question the most. Sucks. I mean, you could say it promotes. You can make an argument for that, but I wouldn't be so sure. Anyway, guys, if you like, here, let me prove it to you. Yeah. Hang on, go back to view. I pretty much just found dark mode word. That to me is amazing. Anyway, if you guys like this video, subscribe. I know I'm repeating myself on like a broken tape recorder, but if you want more video, or if you want like the lab tutorial, or like how I do the lab, or online virtual lab, because this is still coefficient. I don't know what to say. That's a strict word. Like, share, subscribe. I'll make more videos if you comment and help me out. Otherwise, thanks. Bye.